Um, can everybody please take their seats? Okay, good evening, everybody, and welcome to this public lecture by Professor Paul Nurse. It's a real pleasure to have Professor Sir Paul Nurse visiting TIFR, and I'm glad you're all here to hear him. I would like to begin by requesting our director, Professor Jairam Chengalur, to say a few words about the public lecture.
Thank you, Maitreyi. So it's a pleasure to welcome all of you to this public talk at TIFR. And as many of you probably know, research at TIFR covers a huge range of areas, all the way from the largest structures in the distant universe to trying to understand the smallest constituents of matter, and almost everything in between. And in many of these areas, if you look at it, you'll see that TIFR, the areas that we are doing research at TIFR, we are one of the very few and sometimes the only place in the country where such studies are being carried out. So we've always regarded it as part of our mission to have a vibrant outreach program to try and communicate to the public the excitement of doing cutting-edge research. TIFR's outreach program is not just one of the oldest in the country, but it's also probably one of the largest and the most varied. So, for example, at our radio telescope, which is located in rural Maharashtra, we run annually what is probably the largest Science Day program in the country. All of our centers in Bangalore, in Hyderabad, elsewhere have very vibrant outreach programs of their own. And here in Mumbai, we have the long-running Chai and Why series at Prithvi Theatre, which is a public talk which happens every alternate Sunday. So if you're interested in knowing more about our outreach program or in what the next event that we are going to hold, please do check our webpage and our social media feeds. Today, we are truly delighted to host a very distinguished speaker, the Nobel laureate Sir Paul Nurse. You'll probably hear soon a formal introduction of the speaker. But what I'd like to do now is to take this opportunity to thank him for accepting our invitation and being here with us at TIFR. He's also met with our students and interacted with our faculty earlier today. And I'd like to thank him very much for taking time out of his busy schedule to do all of this. Sir Paul Nurse's visit to India is part of the Ashoka University Lodha Genius program. And I'd also like to thank very much Professor Vijay Raghavan and others who've helped arrange this visit to TIFR. So with that, I'd like to stop again and thank all of you again for coming. Welcome again to TIFR.
Thank you, Professor Chengalur. I'd like now to introduce Professor Paul Nurse, and it's a real honor to be able to do that because Professor Paul Nurse is one of the greatest scientists of our time and is also a scientific hero. Many of you here, or at least some of you here, will, I'm sure, know that he is the co-discoverer with Lee Hartwell and Tim Hunt of proteins that regulate cell division. And this discovery earned the three of them the Nobel Prize in 2001. What you may not know, and I learned this from the autobiography he wrote as part of his Nobel Prize, was that he almost didn't make it to university, apparently because his marks in the foreign language section were not adequate for admission. And so he actually started his research in a microbiology lab at the Guinness Brewery before a professor at Birmingham University actually realized or looked at his application and persuaded the authorities to ignore or overrule the poor performance in the foreign language paper. So from university at Birmingham, he went as a PhD student to the University of East Anglia in Norfolk, where he was also born, to Tony Sims' lab, where he actually started work on cell division. And in particular, he looked at amino acid fluxes accompanying cell division in a fungus called Candida utilis. He again wrote in his Nobel biography that he spent a lot of time towards the end of his PhD figuring out what he should do as a postdoc, because he reasoned that if he was going to put in the effort, it had better be to do something important. And he wrote that he felt that studying the cell cycle in a simple organism, in a unicellular organism, might illuminate what is central to life, which is reproduction. And he says that that's what drove him to focus his energies on understanding how single cells divide. And so he's been investigating the regulation of the cell division cycle, which has remained his enduring interest throughout his career, spanning the last 50 years. So he went as a postdoc to Murdoch Mitchison's lab at the University of Edinburgh, where armed with the power of yeast genetics, so genetics in the fission yeast, he used genetic screens to successfully identify mutations or mutants that were defective in the cell cycle. He had this sense, because the fission yeast cell divides on attaining a particular size, he had this sense that he could identify cell cycle defective mutants just by looking for mutants that were either larger or smaller in size than their wild type or unmutated counterparts. And many of you here, at least biologists, will know of the two most famous mutants that came out of this screen. One of them was, of course, CDK, the cyclin-dependent kinase 1. And I'm not going into the details of what it does. The other was V1, which he named because it was isolated in Edinburgh, and it had a phenotype that caused cells to be smaller than their wild type counterparts because they were dividing far too early. These mutants actually helped uncover the rate-limiting steps of the cell division cycle. And then when he moved to his own lab in Sussex, he cloned the mutants, and as I said, identified that they affected the genes CDK1 and another kinase that inhibited CDK1. He moved from Sussex to Lincoln's Inn Field, the Imperial Cancer Research Labs, and then to Oxford, where he became chair of the Department of Microbiology. Followed that with a move to the Rockefeller University in the US, where he was also president. And since 2010 has been at the Francis Crick Institute in London. And many of you will know that the Francis Crick is one of Europe's largest biomedical institutes. And Sir Paul Nurse is both founder, director, and chief executive officer of the Francis Crick Institute. Over the years, his lab's work has been uncovering the intricacies of the cell division cycle and its regulation, but also unraveled how cell geometry intersects with the cell cycle and provided fundamental insights more generally to the regulation of cell size and cell form. He's also been using chemical biology tools to screen for small molecules that could interfere with the regulation of cell cycle as potential anti-cancer therapeutics. So in addition to winning the Nobel Prize, Professor Paul Nurse has also received the Albert Lasker Award for medical research in 1998. He was elected Fellow of the Royal Society, of which he was president for 5 years. And I just read in the newspapers a couple of days ago that he's set to become the president of the Royal Society for a second time in November. And I believe that it's the first time in the history of the society that this has happened. He's also a foreign associate of the U.S. National Academy of Sciences. He was knighted in 1999 for his services to science. And he's received a number of other honors, including the French Légion d'honneur and the Japanese Order of the Rising Sun. He's also advised a number of prime ministers and the European Commission on matters related to science and technology. And to sort of deviate from this entire list of recognitions, I'll also add that I learned that he flies both small aircraft and vintage aircraft and is a qualified bush pilot, which means he can land aircraft where commercial planes don't dare to go. I'd like to add that in addition to his scientific papers, from which you'll see that the experiments are sort of very elegantly done and the science is beautifully written, he's also been a great advocate of the scientific pursuit. And I recommend the young people to read some of his writings on the nature of, doing research. And I'd like to quote him from one of his writings, in which he says, "Research papers are usually revisionist histories, imagined accounts driven by impeccable logic and unbroken experimental success, which in no way reflect the slowness, messiness, and serendipity of real life in the lab." And he's also said, and again I quote, that scientific understanding is often beautiful, a profoundly aesthetic experience which gives pleasure, not unlike the reading of a great poem. He's also a great communicator of science, and he's written a book, What's Life? And it's also the subject of his public lecture today. And so I'll stop here to welcome Sir Paul Nurse again to TIFR and ask him to come and share his thoughts with us.
Thank you. Well, good afternoon. Thank you for that very nice introduction, and thank you all for coming. It's a pleasure to be back in this institute, to be back in Mumbai. I'm on a visit to India, which unfortunately finishes tomorrow morning when I fly back to London. I'm going to talk today about one of the fundamental questions in biology, probably the most fundamental, really, is what is life? It's very easy to ask that, but not so easy to answer. And I ought to admit that I'm not going to actually answer it exactly in a sort of dictionary-like way. But I'm hoping that you will take something away that will allow you to think more clearly about this problem. Let's start with the problem. There's an extraordinary diversity of life on our planet. All around us are bacteria, fungi, plants, animals, and of course ourselves, human beings. So what is it that links all these different life forms together? Well, it's a long time ago, but I think it was a butterfly like this one that first started me thinking seriously about this question. It was early spring. I was around 12 years of age, sitting in our garden in northwest London, in Wembley, where the stadium is, when a quivering yellow butterfly, this species, this one here, flew over the fence. It turned, it hovered, it briefly settled, a shadow disturbed it, it took flight, it disappeared again over the opposite fence. Not a big event, you might say, but that intricate, perfectly formed brimstone yellow butterfly, that's the species, made me think about this problem because it was both utterly different to me, yet somehow familiar. Like me, it was obviously alive. It could move, it could sense, it could respond. It seemed full of purpose. And I found myself wondering, what does it really mean to be alive? And that is, of course, the same question about that is, what is life? The question's been asked by others over the years, and there's been a number of books written with exactly that title. This is one of written, the most famous probably, written in 1944 by the famous physicist Erwin Schrödinger. His book was influential for me when I first read it, nearly over 50 years ago. And he considered one particular aspect of life that he was fascinated by, something he called the code script, and I will return to that. My approach is a bit different, it's a bit wider, and that is to consider 5 of the most important ideas of biology, describing where they come from, because unexpectedly, the most important ideas have generally been around for quite some time, explaining what they are, and then pull them all together to develop a set of principles fundamental to life. And that should give greater clarity as to what life is. Now, the first of these ideas is the concept of the cell. The cell is the basic structural and functional unit of life. I like to call it life's atom, the basic sort of unit of a structure of matter. This is the basic structure of life. They were discovered by Robert Hooke. He drew the first illustration of a cell and published it in 1665. And the work he did was in Oxford. It's a city where I live. There's a plaque on the wall celebrating it. He used a microscope rather like this one here, and he used a razor to take slithers of cork, of plant material, examine them under an early microscope. And his illustration is the black and white one, and he saw neat arrays of boxes. He called them cells after the Latin word cella, which means small cubicle. This is shown in a drawing which he published in a book called Micrographia, which had many, many illustrations of what he saw under a microscope. I've also put up here a scanning electron microscope of the same material. And you see, we haven't advanced very much in over 350 years. It looks remarkably similar. A few years later, the Dutch draper living in Delft, called Antony van Leeuwenhoek, scraped between his teeth and looked at it under a simple microscope, but one that could magnify very well, and discovered single-celled life, bacteria. And he was in correspondence to the newly established Royal Society. He wrote 1,000 letters there, which are in the archives. which I've looked at actually. And this is an illustration from one of them. These are bacteria that he saw between his teeth. I like the charming figure B to C to D. It was obviously swimming around under the microscope and did a little sort of loop the loop, which I occasionally used to do in aeroplanes, as you heard that I fly. I've just stopped, actually. I'm thinking I'm getting a bit too old for this sort of thing, but only very recently, last year, in fact. Now, Antony von Leeuwenhoek was rather disturbed by this discovery of microorganisms, living things in his teeth, because he was particularly proud of his dental hygiene, and it rather disturbed him. And he noted this in his letter that he sent to the Royal Society. Now, he lived in Delft, as I said, and he actually was a close neighbor to the famous painter Johannes Vermeer. And Vermeer was unusual in that he painted two portraits of a scientist. This is one of them. One's in the Louvre, the other's in Frankfurt. This is the one from the Louvre. One called a geographer, the other called astronomer. And I like to think this is Leeuwenhoek. Now, there isn't great evidence for this speculation, so it's probably fantasy. But I will tell you one thing that's of interest. When Vermeer died, he died in his 40s, Leeuwenhoek was the executor of his will. So just maybe, at least in his mind, he had, in doing these two pictures of the scientists, it's the same person, he was thinking of von Leeuwenhoek. Now, over the next 2 centuries, 200 years, it became clear that all living things, all living organisms, consist either of a single cell or a collection of cells, like ourselves. This conclusion was summarized by the German zoologist Theodor Schwamm, most clearly in 1839, when he wrote in German, translated here, "We have seen that all organisms are composed of essentially like parts, namely of cells." So that's the definition that a cell is the basic structural unit of life. But they also are the basic functional unit of life. And that was rather expressed well by another German scientist, Rudolf Virchow, who was the founder of pathology. And he, in 1858, so about 20 years later, wrote, and it's here, "Every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life." That is, it expresses the function of life, and therefore understanding life is all to do with understanding cells. He also said, all cells come from cells. He quoted it in Latin, omnis cellula, all cells, acellular, from cells. Wasn't the first to say this, but he said it in this same article. And this was important because it means cells arise through the division of pre-existing cells. Because it was thought at the time by some that cells just appeared spontaneously in sort of a pond or liquid or so on. But this showed that all cells come from pre-existing cells. And I'm going to return to this at the very end of my talk, but I'll just plant it here so you think about it. This means we are connected back in evolutionary time through successive rounds of cell division that have produced us. We came from the egg and sperm of our parents that fused together, generated by cell division, but it goes back and back and back to the beginning of life. And it's something we don't think about, but it is really quite extraordinary. So cells are interesting. If I fail to convince you of that, let me show you this picture. This is a mammalian egg being fertilized by sperm, and remind you all that once every one of you in this room looked like this. Every one of you was a single cell. So you should think it's important. If you don't, I see the doors at the back are still open, so you can escape now if you want. I should also say cells are considered to be the simplest entities which unambiguously exhibit the characteristics of life. I use the word unambiguously because there are some life forms, viruses in particular, which people argue about. And I'll say something about that towards the end of my talk as well. But what this means is that understanding cells is key to understanding life, so they will figure quite prominently in what I have to say. This is a modern picture, artificially colored, of a cell. They are bounded physical entities, separated from their environment, surrounded by a semi-permeable lipid membrane, which separates them, but allows communication with their surroundings. And it's this separation is quite important because it allows order to be built up inside cells, the order of cells, at the expense of increasing disorder outside cells. And so in this way, life does not contravene the second law of thermodynamics, where everything is degenerating into atomic chaos, as I shall, you'll see in a quote in a moment. And it's this law that often troubles physicists thinking about life, like Schrödinger. This is a quote from the book, which reflects what I just said. And he commented on an organism's astonishing gift of controlling a stream of order on itself and thus escaping the decay into atomic chaos, which of course is where the second law of thermodynamics goes. And he was impressed by the inheritance across generations, the inheritance across every cell division. And he speculated that the reason for that was this code script, which I briefly mentioned. He was referring to chromosomes that were seen in a dividing cell, and he speculated they contained some kind of code script which could explain the entire pattern of the individual's future development and function. And these quotes introduce quite nicely the second idea I want to share with you, and that is the idea, the concept of the gene explaining the mysterious phenomenon of inheritance. Now, this is the idea that the gene is the basis of heredity, and it had its origins with the work of an Augustinian monk, Grigor Mendel, abbot of Brno Monastery, now in the Czech Republic, and he published 2 papers in 1866. He tackled the problem of inheritance by carrying out crosses of pea plants with different characteristics and analyzing the outcome of those crosses in the thousands of progeny that were formed. And I found this picture of his garden where he did these experiments. It was in 1981 when I visited the monastery, right in the middle of the Cold War. It really wasn't quite so easy to get there. What I wanted to point out, this was a large garden. This was a large commitment that the monastery was making to research of this sort. He had a greenhouse of similar size, which had fallen down, but it's being replaced at the moment. And the monastery was committing significant resources to the study of the inheritance from plants. It was like a little mini research institute. And we don't think of monasteries as doing that, but that's exactly what was going on. He used peas, as I said, and by the way, he initially did preliminary experiments with about 6 other species, couldn't understand it, couldn't understand the phenomenon, and did understand something out of peas, and decided only to work on what he could understand. And there's a lesson there that in biology, which is often very complicated, it is useful to work on systems that you can make guesses at what's going on. And if you've then got that right, you may be able to apply that knowledge to systems that which are less clear about it. Anyway, he famously observed in the outcomes of these crosses simple ratios in the characteristics. I'm not going to explain why this comes about. Many of you will have been taught it at school, but ratios like 3 to 1, 1 to 2 to 1, 9 to 3 to 3 to 1, and so on. And this data eventually led to Mendelism, the idea that heredity is based on pairs of indivisible particles he called factors, one of which is inherited from the pollen, and the other which is inherited from the ovules in plants, and in animals from sperm and from eggs. And this was an abstract model, and it fitted the data very well. But when you read it, and I've actually handled the handwritten manuscript of it in the monastery, I feel quite excited about that. But what he was looking at, because he was trained as a physicist, he was very quantitative. Biologists of the time, Darwin actually did crosses and also revealed 3 to 1 ratio, but he made no comment about it, and his data was less accurate. But Mendel's data was very good, and he as a consequence, was speculating about a particle, an indivisible particle. It has to be said, though, that nobody took any notice of what he did. Published in 1865, and it was rediscovered in about 1902 by 3 further plant geneticists by them. It was called, but by that time he had died, so he never saw his discovery, never got to, he never realized how important it was. Now, these factors that he identified were found to be linked to chromosomes, which were discovered after his death. These are, of course, separate in dividing cells, as shown here in this late 19th century of an onion root tip containing cells undergoing division. And in fact, this is the first time I saw cells and chromosomes also with an onion tip squash when I was at school. And why I mention this is because the behavior of the chromosomes, which are a bit like the sort of particles, was the same as what Mendel had described for the segregation of the particles. So this observation had the capacity to turn Mendel's abstract interpretation into a concrete one that you could see. And I'm sure that's the reason why it was much more spread, much more positively received in a few years with the discovery by 3 new geneticists. These factors that Mendel identified are what we now call genes. In the 1940s, Oswald Avery in Rockefeller University, which I worked at myself, showed these genes were made of deoxyribonucleic acid, or DNA. And here is the DNA molecule, which was shown to be a double helix about 10 years after Oswald Avery in London by Rosalind Franklin, Ryan Gosling, and Maurice Wilkins, and in Cambridge by Francis Crick and Jim Watson, who interpreted the experimental data and came up with the model of the double helix. And they famously argued that that structure could explain heredity through the linear sequence of bases making up the DNA. What you see here is a cell, a nucleus, and the chromosomes. If you now pull these chromosomes out, you see the beginning of the double helix appearing. And this double helical structure is generated by pairing between complementing bases, with adenine pairing with thymine, that's A with T, cytosine with guanine, that's C and G. And you get a double helix. This is the double helical structure. It's best if you think of it as a twisted ladder, and then in your mind, untwist the ladder. So it now is a ladder. And then each of the rungs has got either a G and a C or an A and a T. If you now cut the rung in the middle and pull it apart, and then just build a new other side of the ladder, Wherever there's a G, there will be a C. Wherever there's a T, there will be an A. And if you follow that through, as is shown here, you'll get a new strand built on the original strand, and this will completely replicate what is seen. There's two strands as are completely replicated, so you have two copies instead of one copy. And this is the account of how heredity works. And the second thing that's important, and which was pointed out by Watson and Crick, is that the order of the bases in the DNA could provide information, and that information could account for the heredity and account for Schrodinger's code script, because it's precisely copied every time a cell divides. Crick, who was a very imaginative man, went on to propose what he called the central dogma, because he also wasn't a very modest man either. He was imaginative and not modest, and it was a central dogma, most important thing, which explained how DNA encodes the structure of proteins. Now, the central dogma describes how the sequence of bases making up a gene is copied from DNA to ribonucleic acid, RNA, that acts as a messenger from the DNA located in the nucleus, where the chromosomes are, a message from there to the cytoplasm where proteins are made. And there's only one gene encoding each protein. Now, these discoveries laid the foundations for molecular biology, so really critical. They're critical for understanding life. Central to it is the genetic code. It's written in the four-letter code of the four bases, and the information is stored in a linear form. I want to emphasize that. This way of storing information and communicating information is the same as the words I'm speaking to you. That's a linear stream of words. It's the same as words you would read in a book, which is a linear sequence of information. And it's the same, of course, as bytes in a computer, which are also stored in linear form. And what it means, of course, is that the linear digital storage of information was invented by life over 3 billion years before the computer age. We came on the scene a bit later. Now, the nature and function of the gene introduces us to the next two ideas, life as chemistry and life as information. Let's look at life as chemistry. How chemical activity and also physical forces can bring about life. Now, the origins of this idea can be traced back to Lavoisier, the father of chemistry. He was studying fermentation of wine. He was French, of course, so obviously interested in wine. Unfortunately, he was also a tax collector for the Ancien Regime. You can see why I failed French O-level so many times. And as a consequence, he lost his head on the guillotine. So that did, of course, stop his research prematurely. But it was taken up again by Louis Pasteur, also French, shown here, who carried out experiments on sugar beet fermentation, which led to him stating most clearly the concept that life was chemistry. What he was doing, he was living in northern France at the time, he was helping industrialists who were analyzing the fermentation of sugar beet, which is used to make alcohol, because quite often those fermentations failed. And he looked at a failed fermentation and a successful one, and the successful ones always had yeast in them. The failed ones didn't, but he thought had bacteria in them. So he proposed that yeast cells produce substances that make alcohol from the sugar, derived from sugar beet. But if yeast wasn't there, bacteria could grow and they produce acid instead of alcohol. So this was very useful to the industrialists, because what he said is if you put yeast in there, it will always work. I don't know if he got paid for this or not. But it certainly was useful. But he was a scientist. He was interested in what it meant. And he concluded more generally that fermentation is a physiological act yielding products for the cell. So chemical reactions are an expression of the life of a cell. In other words, life is chemistry. But it is a very elaborate and special chemistry. It's based on carbon polymers, on protein carbon polymers, but DNA and RNA are carbon polymers too. And I just want to now discuss them, because the substances from yeast that Pasteur postulated made the alcohol are proteins acting as enzymes that catalyze the chemical reaction producing the alcohol. Proteins are polymers of amino acids. They have a backbone consisting of carbon-carbon, nitrogen, carbon-carbon, nitrogen, and then, which is part of each amino acid. There are 20 different amino acids, and these amino acids form side chains, and you see some illustrated here: lysine, leucine, tyrosine, aspartic acid. And what's important about these side chains is they are chemically different. Some are acidic, some are basic, some are bulky, some are not bulky, some are hydrophobic, don't like water. And these are examples of the 20 amino acids that make up proteins. And the result of this is, unlike DNA, which is chemically boring, proteins are actually interesting chemically because of this variability in the chemistry that is there. This is showing another picture of the bond. You can see the CCN, and coming off it are the R's in yellow there, which are the amino acid side chains. Now, these amino acid side chains influence how a protein folds up. Because what you see here is a chain of amino acids of a protein. They are chemically interacting one with another to produce a particular three-dimensional structure of the protein. And this generates something like this, which is a billiard ball model of protein, where the different chemistries that are around are coded by different colors. So you can see proteins can produce very elaborate, three-dimensionally, chemically diverse structures capable of doing exquisite chemistries that are the basis of life. So enzymes act as chemical machines. These machines produce the huge range of chemicals that living organisms need. They make new molecules. These enzymes, they break molecules down, they recycle them, they generate molecular assemblies, which can synthesize the polymers of protein, nucleic acid, lipid, carbohydrate. They can do physical work. They make motors, which carry cargoes around on the cell on protein-based trackways, just like some little railways. They act in hybrid ways, combining chemistry and physics, capturing energy from the sun to produce energy-rich chemicals that can drive all these activities throughout cells. So all this activity means that there are thousands of chemical reactions being carried out simultaneously and close to each other in living cells. A small sample of these are shown here. Each of these dots, this is a map of central metabolism of a cell. There's several hundred reactions going on here, which is only a fraction of what goes on. And I had to learn what these dots were at university. For those of you, thank goodness we don't have to do it anymore. At least I don't think we do it anymore. In the middle there, you'll see the red circle, which is the Krebs cycle. Now, for each of these dots to be successful in a chemical reaction, they sort of have to be a bit separated from other dots because the chemical conditions can vary from one to another. And that's achieved by compartmentation, brought about by a variety of mechanisms, the surfaces and folds of the enzymes, so the protein makes sort of cavities where things happen, complexes of several enzymes or proteins acting together, and membrane-bounded organelles, which is really what we're looking at when we look at a cell as schematized here picture I think I stole from Scientific American. All of these, you look at this, we think of structure, but really I'd like you to look at it and think each of these are reflecting the different chemical microenvironments, which allows the hugely elaborate chemistry of life to take place. It is amazing in 2, 3, 4 micron sort of cube cell that all these reactions can occur successfully. But it is not enough that they can occur. They also have to be organized together. And that is a second and very, very big problem, because they have to be organized together. That means there needs to be communication to the different components and the different chemical environments. The communication is one of the first steps of information, the management of information. And that leads to the next idea I want to speak to you about, which is life as information. So I've spoken about life as chemistry and, of course, physics, life as information. This has always been around. In fact, of course, Mendel's work is basically said this, but it's becoming more to the fore, and I think will be important in the immediate future. It touches things like control, purpose, and complex systems. Complex systems, where we have to think about how a complicated system actually works together, is a characteristic of life. And I wanted to find where this was first discussed. And to my surprise, I found it could be traced back to the philosopher Immanuel Kant, writing at the beginning of the 19th century, where he argued that what goes on in life are complex systems. He wrote 20 pages on this in a book on moral philosophy. I have no idea what the relevance it was to moral philosophy, but it really was quite insightful, written about 1800. So the operations of complex systems themselves are based on being able to manage information. And I want to give 2 examples of this to illustrate it. The first you've seen already is the structure of DNA. Now, the structure of DNA is iconic and it's beautiful, but its real beauty lies in the fact that it only makes sense when it's recognized as a digital information storage device. So forget about the structure. It's actually an information storage device, the one written, as I've said, in the linear script of 4 letters. Now, DNA is ideal for storing information because the encoding letters, that's the yellow rings here, are turned inside the helix and protected from chemical change. But this information can be turned into chemical action that can do work through its encoding of protein. And for me, this is a wonderful characteristic of life built on carbon-based polymers. The ability to encode information in a one-dimensional structure of DNA, which is essentially largely chemically inert, and yet to turn that information into chemical and physical work in chemically active 3-dimensional structures, which is separated. And that is something I will briefly return to later, but we don't generally think of it like that. But that's what's happening. And it will be important in thinking about life on other planets. Second example about information I want to tell you about is concerned regulation. That is making complex systems act as a whole with purpose. Now, a familiar example of regulation is most easily seen usually in man-made machines because we designed them. If you're like me, I only understand machines built before about 1900, and after that, they increasingly become obscure to me. But this is a governor from a steam engine that I discovered in a boat, and you can see how it works. The spindle is rotating, the balls are swung out, okay, and that lifts up a valve and shuts off steam going into the steam engine. It slows down, the balls go back in, and now you can now change the valve position and put steam back into the engine so it can speed up again. A governor was developed by James Watt to regulate steam engines. Now, this is an example of a negative feedback loop. Now I'm going back to life from our man-made model. Negative feedback loop. We've got a substance A turned enzymatically into B, and enzymatically B to C. And now imagine that C inhibits the enzyme between A and B. And what you can see, what will happen is, as C builds up, it inhibits A to B, which stops C being more synthesized more, and it leads to homeostasis. Positive feedback, the next one down, is completely the opposite. As C builds up, it promotes A to B, so it turns into an irreversible switch. This type of logic was first properly demonstrated by the French geneticists François Jacob and Jacques Monod, entirely based on logical analysis of gene regulation of sugar metabolism in bacteria. No biochemistry, no chemistry, just the logic of what they were actually observing. Now, many such feedbacks and control modules operate in cells. They can maintain homeostasis, as I hinted at here, generate switches, as here, timers, toggles, oscillators, all translating chemistry into informational modules. They can be linked together to generate even more elaborate routines of informational management and processing. A metaphor for this is an electronic circuit. This I stole from a journal, scientific journal. It's an electronic circuit. And what the person who wrote the article has named different components in there after certain oncogenes like RAS and SARC and so on. And he's trying to say that if we want to think about cancer, because they're all involved in cancer, then we should think of a network of connections that is important for it. But an even better metaphor for some living systems is wetware rather than hardware, because actually the way cells work is through solution chemistry. You connect from one component or one pathway to another through solution chemistry. And that means you can re-link and reconnect, increasing the versatility of the control modules. So you're not just relying on the output of the network is set up. The network can change, and we barely begun to understand how that works yet. And I think it will be important. Now, the management of all this chemistry by information and allows the behavior of a complex system to act as a whole with overall purpose. I'm deliberately using that word, it's a bit challenging. And by overall purpose, I mean you reproduce a cell, you maintain the metabolism, you make a butterfly, or we ourselves are kept alive. Now, this purposeful behavior for centuries was thought to require, for its generation, a designer, a creator. And it took evolution by natural selection to change this, and that is my final idea. Evolution by natural selection, how living things come about. Now, I can tell you, we're getting towards the end of my lecture. So if you're getting a bit tired, just take a big breath now, and it'll be all right, okay? It'll be all right. Now, this evolution by natural selection, it is a beautiful idea, mainly due to Charles Darwin. The idea has two rather separate parts. The first is that life evolves over time. And the second is a major mechanism for it, for evolution, is natural selection. Now, the first of these, that life evolves, was not Darwin's idea. It had been talked about for 70, 80, 90 years previous to him. And in fact, one of the people who spoke quite a lot about it was Erasmus Darwin, who was Charles's grandfather. Now, you can see Erasmus on the left there, and Charles, a rather stern-looking Victorian gentleman, on the right. And Erasmus and others of his time, like Jean-Baptiste Lamarck in France, really discussed life evolving, but they didn't have a good idea of the mechanism for it. I want to say a few words about Erasmus because he's not so well known and I'm rather fond of him. He was a colourful character. He was a scientist, but he's earned his money as a doctor, but he charged his patients according to whether they could afford to pay him. So he charged his rich patients quite a lot and treated his poor patients often for nothing at all. He was a poet, and he wrote up his research, most of it, in the forms of verse, poetry. I managed to buy a number of his books from the time. He wrote in 1790 to about 1805, and it's full of this verse describing various scientific things. I mean, it's the weirdest thing. It's quite colourful and so much more interesting than reading scientific papers today, of course. And it was simply odd to see this. He was quite famous as a poet. He was a Republican and didn't agree with the monarchy. He refused to be George III's doctor because he was a Republican. And unexpectedly, he was, because you wouldn't know this, most people don't, he was a proponent, the first proper proponent of women's education in public schools. And so much so, he wrote a little book about what girls should learn. And his two daughters set up a school teaching girls. I mean, amazing, some of these people. He was also a great conversationalist, despite having a terrible stammer. And he was absolutely nowhere near any university. He was working away as a doctor in the Midlands. Now, that life evolves over time has an implication. An implication is that there is a tree of life, that living organisms are related. I just want to touch upon that. Because if life only arose and survived once, as seems likely given the conserved chemistry and coding, then all life on Earth is related to each other. And to state that another way, all life that we see around us are our relatives. Some of them are very close relatives, you know, like a gorilla. Some of them, like my yeast, I do research on yeast, is much more distant. This connection was caught by the only illustration in the famous Origin of Species, published in 1859 by Charles Darwin. And what you can see here, he's constructed a tree. And on the top line, we see the species that have survived. And in between, we see this network, including many that have gone extinct. I want to refer you to the bottom, A to L, because he didn't know whether life appeared once or many times on the planet. This suggests many times, or a number of times. What we would say now today, because we understand the chemistry, which he did not, that it is likely, I would say, that it arose either once, or if many times at a very early stage, only one actually survived. Now, Darwin gathered evidence in favor of life evolving, and particularly through natural selection, when he went round the world for 5 years as on the Beagle, which was a Royal Navy British boat mapping South America coastline. Now, I want to describe his idea of natural selection very briefly. It's a consequence of a population of species exhibiting variations caused by inheritable genetic changes. And of course, we've talked about that, which can be caused by mistakes in the DNA when copied or damaged. If these variants influence characteristics that can make these organisms more successful and more adapted, then these will reproduce more. And therefore, this particular variation will spread through the population. And over time, such genetic variations will accumulate and lead to changes in the population such that the individuals of the new population will no longer be able to breed with the original population, leading to a new species, which is why it's called the origin of species. A new species which is better adapted and more successful in survival and reproduction. So Darwin came to this conclusion in part by his studies of finches on the Galapagos Islands. On the left are the illustrations of the finches that he looked at. On the right are pliers that have been added, okay? He didn't look at the pliers. The one that the finches, the one at the top breaks nuts. The one at the bottom finds insects under bark. And the Galapagos are volcanic islands, haven't been around there very long. It seems that a finch got there, a sort of prototype finch, and then evolved here into different lifestyles and became modified for it, an evolution by natural selection. On the right, we have a family of pliers which do different jobs. They're clearly related, but they've been designed to do different jobs, as you can see looking at them. And I put this up there because on the left, we have evolution by natural selection. On the right, we have intelligent design. And this is the debate that still is quite contentious for some who feel that this variety of life has to have been intelligently designed, whereas biologists would think that it can appear by natural selection. Now, for natural selection to take place, three things are needed: reproduction, of course, a heredity system, and a heredity system that exhibits variability upon which selection can be worked. And that's related to the ideas of the cell and the gene. So let's just look at that. Imagine a single-celled organism. Imagine that it's got a brown coat and it could have a red coat, that change is decided by a single gene, and that they can mutate. So we have a yellow cell, there's a mutation, and that leads eventually to a red cell. Also imagine that red cells are not nice to eat, whereas yellow cells are. So if you're a red cell, it's an advantage. And over time, red cells will come to dominate in the population. So the idea of the cell and reproduction combined with a genetic system which exhibits variability is the basis of natural selection. It's a very important idea, beautiful idea and important. So much so that the 20th century geneticist Hermann Muller used it to define what life is. He defined life as living things have properties which allow them to undergo natural selection and therefore to evolve. And I'm going to return to that in a couple of minutes. But before I do that, I firstly want to say goodbye to Darwin. This is him as an old man, and I want to read out to you his wonderful final monumental sentence from The Origin of Species. Whilst his planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Isn't it a wonderful sentence? I'm back. You know, when I write a paper, I can't bear to read it after a month. You know, it's gone through editorial review. Don't speculate about things you're not sure about. It's been reviewed by people taking out words because it's too fancy or whatever. And it's just dry. We can still read these words that were written in 1859 and be excited by them. There's something else I want to point out about this quote. Notice he refers to gravity. Now, what on earth has gravity got to do with this? He's referring, I think, cunningly to Isaac Newton and the law of gravity, because the physicists, the queen of sciences, build their subject on laws, and whilst biologists sort of collect postage stamps, bits of sort of information. That's not my quote, it was a physicist who said that. And what Darwin's saying here is, I have a law here which is just as powerful as laws in physics. And of course, it is more difficult to produce such laws in biology, but they are really important that we do it. Finally, I have to say something about viruses, and then I'm going to sum up, okay? Now, viruses have a genome. You know, viruses are what you have to write school essays about. Are they alive or are they dead, right? And I'm going to give my answer to that question. Viruses have a genome based on DNA or RNA. They undergo evolution by natural selection. Therefore, they pass Muller's test. You remember Muller? But they can only reproduce when they are inside the cells of other host living organisms. They do that by hijacking the cell's molecular machinery. So it will copy the virus and make genome and make more viruses. This means that a virus is completely dependent upon another living entity, which is the reason why, is it alive or is it dead? But in thinking about this, it's important to remember that other forms of life are also, to greater or lesser extent, dependent on other living things. The many parasites that live on or inside the cells of bodies of animals and plants or fungi are examples of this, although the extent of their dependency is different. We all eat food which is derived essentially and mostly from other living things. We have bacteria in our gut making amino acids that we cannot make. So what this means is there's a graded spectrum of living organisms which reflects their dependency on other living organisms. In the case of a virus, it's very strong. In other organisms, it's much weaker. But these different life forms are all dependent in various ways on others. And so I don't find the question about viruses quite so extreme as others do. It's an extreme dependency, but we all have it, that dependency. And what it, of course, means is that life on Earth is fundamentally connected through these deep relationships and interdependencies, and also through evolution, because life is all related one to another. Now, I began by saying I'd take these ideas and use them to establish principles to understand what life is, and I'm now going to do that. But as I warned you at the beginning, it is not a short dictionary definition. What is life? Well, living things are independent, bounded physical entities. Basic unit of life on Earth is the cell, can't speculate elsewhere. Cells are separated from their environment, but they're in communication of their environment and therefore can generate order and organization. And multicellular organisms like our cells are just aggregates of cells. 2, and this is probably the most critical one, I'm following Muller, living things possess the ability to evolve by natural selection. To evolve, though, they must grow, they must reproduce, they must have a hereditary system which can exhibit variability, and that is the stuff upon which natural selection can actually work. Living things are chemical, physical, and informational machines, and they use that to construct their own metabolism, which maintains themselves, allows them to grow, and allows them to reproduce. That's why I said we couldn't use it in a dictionary definition, because it would take up half a page of the dictionary. There's other things we can add which are not quite so central. Life on Earth is based on carbon polymer chemistry. They produce nucleic acid information storage devices and proteins, which are chemical and physical machines. It's based on information management with storage in a linear form, encoding the polymers. And living things on Earth are related by common descent and are closely interconnected and dependent upon other living things. And these are other things that we need to be thinking about. Let's have, before I end, a speculation about whether we will ever encounter life elsewhere in the universe and what would it look like. I suspect it will be based on similar fundamental principles. The chemistry may be different, may not be built on carbon, it could be built on silicon, for example. That's what science fiction writers often sort of talk about. But to satisfy the absolute need, if you're going to evolve by natural selection, for long-term information storage, I think the chemistry must be built on polymers, because I can't think of any efficient way of storing information or lots of information except for polymers. And just to return to it, the beauty of the DNA encoding proteins is you have long-term information storage being turned into hugely varied chemistry, which can be unstable chemically, but does work, which DNA generally doesn't. RNA is a little bit in the middle. It can do some chemistry, not very well, and it can be also act as a storage device, but not as well as DNA. When we look for life on the other planet, like NASA, we always look for the chemistry and disturbances, because we can't actually consider what I just said, but it just maybe we might miss if there was a different sort of chemistry. Okay, but let's come back to our planet, because it's the only corner on the universe, where we know for certain life does exist. The life that we are part of here is of great diversity, and we are making sense of it. This understanding, growing understanding, has great potential, improves our culture and civilization, but also the lot of humankind. But it does go rather further than that. Biology shows us, as I've emphasized, that all living organisms we know of are related and they closely interact. And that means ultimately we're bound by rather a deep connectedness to all other life, to bacteria, fermenting yeast, pea plants, and back to my yellow brimstone butterfly. They've all been part of this talk. Together, these species are life's great survivors, the latest descendants of a single, immeasurably vast family lineage that stretches back in time through an unbroken chain of cell divisions, in fact, to the far reaches of deep time. I said that earlier and I'm repeating it because it's extraordinary. We somehow have this connection going back through billions of years connecting us. As far as we know, though, we are the only life forms who can see this deep connectivity and can reflect on what it means. And I argue that gives us human beings a special responsibility for life on this planet, made up as it is by our relatives, some close, some more distant. We need to care about it. We need to care for it. And to do that, we need to understand it, which is why I've given you this talk. I've also written it up, which, if you can get it, is in this book, What is Life? Thank you very much for listening to me. Not sure what I do next, do I? I'm happy to do questions. I'm deaf, so you have to repeat them to me, okay? Can you do that?
Questions.
Hi, Paul. Nice talk. So, would you call self-reproducing machines, man-made machines, which have a degree of randomness to allow for like hereditary changes, as alive?
Not if it's, I wouldn't unless it's set up to evolve by natural selection, because it cannot then adapt and optimize, and we cannot get overall purpose emerging. The beauty of the evolution by natural selection is that you end up with an increasingly perfecting system that can grow and reproduce. If you just sort of have something which can't do that, it's static. So that's why I would say I don't think so. Now, having said that, something had to arise first before going through all those processes. And that's the origin of life. And if you're going to ask me, how did life originate? I'm just going to say, I don't know.
Yeah, thank you.
Thank you for the question, and very clearly asked.
So good evening. My question is, prokaryotes have got the double helical structure, but they lack the telomeres, which is primarily because of the quadruplex structure, which is seen in eukaryotes. So, could this be a very important aspect of the evolutionary process? And a very weird question, which I'm going to ask is there a possibility of 6 helical structure and more also?
Not clear I'm getting the question. You
I'll
said
repeat
the
it.
helical.
I'll repeat it again.
Yeah,
See,
just the helical. I mean, the helical bit. I
the
didn't.
quadruplexed DNA and telomeres are very specific to the eukaryotes and not to the prokaryotes. Okay.
Do you mind just doing an interpretation? Because
Yeah.
I'm missing too many words. Okay.
He's talking about quadruplex DNA and telomeres being specific to eukaryotes versus prokaryotes.
Oh, between eukaryotes and prokaryotes, yeah?
Yes, yes. And further, which I am asking is that could this be a very important aspect of the evolutionary process?
What do you mean the change between prokaryotes
Yes,
and eukaryotes?
yes.
Yeah.
And could there be
look,
additional helical structures possible in the future, like 6 helical structure, 8 helical structure, 10 helical structures, like?
Well, let's start with the prokaryote eukaryote thing. Prokaryotes are just like eukaryotes with everything I've just said. I mean, where they're different is to do with details of the cellular structure, particularly DNA, which we now have folded up in chromatin and so on, as you were saying. But I think they are essentially similar. Are they subject to different rules in the way I was talking? I'd say no. I'd say everything that we talked about, you know, the gene, metabolism, life as chemistry, that is, life as information, evolution and natural selection, all the same. So I don't think it's a fundamental difference. Eukaryotes can give rise to much greater diversity, so that's a very important aspect of it. But the fundamental property of life, I think, same in both. I hope that's answered the question. Thank you.
Hello.
Hello.
So, in your talk, you mentioned that life arose just once as opposed to multiple times.
Well, I suggested it. I wouldn't like to be confident to say that it did. Well, the reason I say it is that the genetic code is remarkably similar in all living things. And I can only easily imagine that if it had a fundamental origin and it remained unchanged. So if we had a different coding system, I would say it could have arisen more than once, but it is the same coding system, basically. So that's why I speculated. Of course, this happened 4 billion years ago. So, you know, who knows what was going on then? But that's why I say it. Okay?
Thank you for a very comprehensive talk. That was really a pleasure to listen to. Something about your talk made me think of this. When you say multicellular organisms are merely aggregates of single cellular organisms,
Yeah.
just some cells in a petri dish do not spontaneously become a single organism. So I was wondering, do we understand where this transition happens?
Well, I think you're asking about the transition from a single cell to multicellular. And I have a few things to say about that, actually. The first is this, that the first fossils that we see are 3.5 to 3.7 billion years old, roughly. The solar system, the Earth formed, they think, 4.4, 4.6 billion years ago, cooled down over the next 200 or 300 million years to maybe 4.1 billion years. So it means the first and essentially unicellular life only took 400, 500 million years to evolve. Now, of course, that's a huge time. But then for multicellular life to be common, didn't occur till around 600 million years ago. So almost to get life only took 500 million. To go from a single cell to a multiple cell took 3 billion years. And I wonder whether we're missing something. I don't have an explanation for it, but I think we need to think about it. Once we had multicellularity, then we end up with everything we have around us. And that was pretty quick too. Why it took 3 billion years to go from a single cell to multiple cell? I mean, you can speculate that maybe the chemistry, the environment didn't allow larger organisms to live. That might be an explanation. But I'm slightly reluctant to say it because I think it's... so that's my main statement to it. But in understanding what life is, I think I see a fundamental difference between single cell and multi-cell. Certainly, they both are undergoing evolution for selection, which is perhaps key to it. Okay?
Good evening, sir.
Which one? Where? Oh, yeah.
Yeah.
You're in front of it.
Sir, on a cellular level, how is consciousness related to life?
Consciousness?
Yes. And my second question is, what is the motivation behind life? Why all these activities happen?
I would ask a philosopher, I think. I don't understand what consciousness is, even, let alone how it distinguishes us. You can ask does a chimp have? Is it of the? I'm sorry, I seem to be going in and out. Don't have really much to say about it. I actually confine myself to the simplest problems of life, single cell, and I'm nervous to go to consciousness. There will be neurobiologists in the room who will be delighted to talk to you, I'd say. But whether they will really be able to explain it,
Sir Paul, I wanted to know whether human beings have stopped evolving. Have we broken that evolution by natural selection chain? And if we have, then how is that explained by the evolution theory? Because we seem to have all the qualifications to evolve, but are not. And if we are evolving, then in what way are we evolving?
Right, I didn't get that. So could you interpret for me?
Have we stopped evolving or are we still evolving?
Evolving. Okay, that's when I get one sentence, I usually can get it. Okay. Well, what do we mean by evolving in this context? People say this because they say that we've altered the selective pressures. You know, we don't let people starve to death. We cure them of disease. And this somehow is going to damage our genetic makeup such that we are no longer going to be worthy of survival. Okay, I think that's the sort of extreme case of it. I would say the greater danger to the human race is if we didn't worry about people who were ill or thing. And I think that is the way to think about it. We now live in a society, and for our society to survive, we will have to help each other and so on. And that is a much more powerful issue, I would say, than whether we can run slightly faster or not, and we've sort of lost that. There is something else to say, which wouldn't mind, this is beginning to sort of fade, which is, do not make the mistake of thinking that living things as we... And that's really important, because sometimes people make an argument, you know, everything is very efficient utilization will happen that will disturb metabolism, so it's less efficient. We are basically very inefficient. I'm actually losing. Can you still hear me? But is something happened here? Or yeah, maybe I'll use this. Is that okay? Good. Oh, that's much better. Just don't make the mistake of thinking we're perfect. I mean, anything's perfect. It just is better than what it's competing with. Okay, so there is room for evolution, of course, and if I can push it a bit further and try and answer the question a bit more sensibly, I'm just sort of thinking about it as you asked me. The real problem is when the selective pressures change rapidly. And that's what the real problem is with climate change and biodiversity, because it's changing too rapidly to get adaptation to work, because we don't give it time to actually evolve. So this is why the climate change debate is so important. And when people say, oh, we've had great changes of, glaciation and so on, all true, but the changes have taken thousands of years, which allow some adaptation. When it happens in tens of years, then this is a great struggle for our ecosystems and biodiversity. Thought I'd just say that. Good.
Hello, sir. Indian philosophy says that via meditation, we can slow down the mutation and even prevent cancer. So can you please explain that scientifically, how it would works?
I might need again, can you just repeat it quickly for me, somebody?
So how can we communicate with cells?
Look, give it to, just, can you repeat the? Whether what can help? Oh, whether medication helps cure cancer. Oh, you're right. Sorry, I just hadn't said much about cancer or medication. Yes, it will, but it doesn't solve everything. Oh, meditation. Got it. I should have guessed where, given India, of course, but well, it's interesting because I think a few years ago I said no. It might make your quality of your life better, but no. But I've got somebody I hired at I run an institute, the Crick Institute, who is looking at cancers where quite often the nervous system is innovating into the cancer. Now, I didn't know about that 10, 20 years ago, but now that provides a link between the nervous system and the brain, that's a consequence of that, and cancer. So it may be that something might come out of that, which is relating some of these things that you're mentioning, like meditation, just might actually have an explanation. If you'd asked me that 10 years ago, I'd have said no. Today I say possibly. Okay.
Good evening. So just one question. I am Dr. Prashant. I am a psychiatrist. So I wanted to understand whether the behavior as an emergent phenomena emanates from the cellular crosstalk, as we are increasingly moving molecular in psychiatry. So I just wanted to understand what's your perspective on that.
I don't think I have much to say about it really, because I'm not a psychiatrist. I don't think about. I'm an amateur at it. What you should take away from here is really the single cell, which is the simplest living thing. Thinking about the simplest living thing gives us the basic principles for life. It doesn't tell us about human nature. Human nature is a different set of issues which are influenced by that, which have got to do with higher brain function, interacting with others, and so on. And what I've said doesn't have too much to do with that. What it is providing is the platform for these other things to happen. I know I haven't answered the question, but that's what I'd say.
I was just intrigued, like one of the earlier questioners, that it appears that there was only one ancestor to.
Well, maybe.
Yeah, but that's what the genetic, code
say,
to be telling us, which is a little surprising because you would have imagined that multiple things should have evolved since, why did it pick this particular? One would imagine there were many possibilities and many places on Earth which were suitable to life. And so therefore many of these possibilities should have been realized. So I was wondering whether many of them were indeed realized, but one of them actually was much better than the others. And that is the one for which, for example, you have fossil evidence from 3.5 billion years ago, but maybe these things actually started much earlier And fossil evidence only finally happened when they were numerous enough, and that was the one which finally won in the competition.
I completely agree with that. The actual sentence I said is that either only arose once, or if it arose more than once, it's the only one that survived. And I think it's exactly what you say. So maybe there were different genetic codes and so on, but it wasn't so efficient and effective. Yeah.
Thank you. Excellent talk, sir. You mentioned about speciation. So speciation.
Yeah. Are you an evolutionary biologist?
No, I'm not.
Good. Thank goodness for that. I might be able to. Yeah.
So has something like that been observed in a lab in the lower forms of eukaryotes?
I think so.
Through mutation.
Yeah, I think so. It's been observed with my artificial microbial selection. I would say these days we get, of course, with short generation time with microbes, half an hour to an hour, you can do many, many generations, put them under different selective pressures. And I think that's led, if we've got to define a species, of course, and if we define a species by the inability to mate with the original population, then I think I'm confident in saying that that has been done. Yes. And artificial selection, you know, for domestic animals and so on, has for sure occurred. So if you take a dog has arisen twice from wolves. They do not mate with wolves anymore, and they have been artificially selected. It's not in a lab, and it happened fairly rapidly. It only started 15,000 years ago. So that's another example. And domesticated animals and plants have been are the experiment for not that purpose, but that's definitely been observed. Well, in fact, we live in it. I mean, you know, when you eat a sandwich, you're eating wheat, and it came from just a grass seed looking thing. Okay, are we done? Thank you. Thank you for the question.
I'd like to add that there's tea outside. Get out of the auditorium and just follow the crowd. There's tea, and snacks.